All right, so today we're diving deep into the world of TRP channels.
Yeah, these are amazing little proteins, really.
They're like our cell's sensory system, right? Like
Yeah.
tiny little antennas picking up signals from the environment.
Exactly. They allow cells to respond to all sorts of stimuli.
And from what I've read, they don't just react to one specific thing, right? It's
Yeah.
like they have multiple senses almost.
Yeah. That's why we call them polymodal. It means they can be activated by a whole range of things.
So, like, what kind of things are we talking about?
Well, one TRP channel might react to a change in temperature, like hot or cold,
Okay.
but that same channel might also respond to a specific chemical
Oh, interesting.
or even to a change in osmolarity.
Osmolarity.
Yeah, that's the concentration of solutes in a solution. So
Ooh.
basically, how much stuff is dissolved in the liquid surrounding the cell.
Okay, so it's like a sensor for the cell's environment in a way.
Exactly. It's how the cell knows what's going on around it.
So how does this actually work in real life then? What's an example of one of these TRP channels in action?
Well, think about that burning sensation you get when you eat a chili pepper.
Oh yeah, everyone knows that feeling.
That's not because the pepper is actually hot, though.
It's not.
Nope, it's because of a chemical in chili peppers called capsaicin.
Ah, so the capsaicin is what's triggering something.
It triggers a specific type of TRP channel called TRPV1,
TRPV1.
which is found on pain-sensing neurons. So when capsaicin binds to TRPV1, it sends a signal to the brain that we interpret as a burning sensation.
So it's tricking our brains into thinking there's actual heat.
In a way, yes. It's hijacking the same pathway that's normally activated by high temperatures.
That's wild. So are there different types of these TRP channels then? Because you said TRPC1 is activated by capsaicin.
Oh, absolutely. There are actually many subtypes of TRP channels grouped into different families.
Okay.
Like we have TRPV, TRPA, TRPC.
Each with their own specific triggers.
You got it. Each family, and even individual channels within a family, have their own unique set of sensitivities.
So TRPV1 is the one for heat and spiciness.
Yeah, that's the one that chili peppers activate.
What about the other ones?
Well, for example, there's TRPM8,
Okay.
and that one is activated by cold temperatures
Interesting.
and also by menthol.
So that's why mint feels cool and refreshing.
Exactly. It's activating those TRPM8 channels.
So it's not just about the type of stimulus, whether it's temperature, chemical, or whatever.
Right.
It's also about which specific TRP channel is being activated.
Exactly. Different channels respond to different things, and that's what gives us this incredible range of sensory experiences.
That makes sense. Otherwise, we'd be constantly bombarded with sensations.
Imagine feeling pain from the slightest breeze, right?
Or tasting every single molecule in the air.
Yeah. Our senses would be totally overwhelmed.
So it's this diversity of TRP channels that allows us to experience the world in such detail.
Exactly. It's a beautiful system.
Speaking of diversity, where are all these TRP channels located in the body? I mean, are they just on the surface of our skin, or are they deeper too?
They're definitely found in our skin, that's for sure. They're what allow us to feel heat and cold and touch and pain.
Right.
But they're not limited to the skin. They're actually located in various tissues throughout the body.
Oh, wow. So they're kind of everywhere.
Pretty much. You find them in sensory neurons, for instance.
Which are?
The cells that are responsible for transmitting information about the environment to the brain.
So they're like little messengers relaying information from the outside world to our brains.
You could say that. And they don't just relay messages about touch and temperature either.
Mm.
You also find TRP channels in our mouths and noses.
So they're involved in taste and smell.
Yeah, they play a big role in those senses. And
Don't leave me hanging.
they even play a role in vision.
Wow. Really?
Yeah, they're pretty amazing little proteins.
We've got these TRP channels all over our bodies, helping us sense the world around us.
Exactly. And they're not just involved in pleasant sensations either. They're also crucial for something else.
What's that?
Pain.
Pain. Right, because you mentioned earlier that TRPV1 is activated by capsaicin, which causes that burning pain sensation.
Right. And that brings us to a really important concept, nociception.
Nociception.
Yeah, it's basically the process of detecting and transmitting pain signals.
Okay.
And TRP channels are essential for this process.
So if I stub my toe, for example, certain TRP channels are activated and they send that ouch signal to my brain.
Exactly. When a TRP channel on a nociceptor,
A nociceptor.
sorry, that's just a fancy word for a pain-sensing neuron.
Got it.
So when a TRP channel on a nociceptor is activated by a potentially harmful stimulus, it sends a signal to the brain that we interpret as pain.
So these TRP channels are like our body's alarm system, warning us about danger.
Exactly. They're essential for protecting us from harm.
But what about chronic pain? I mean, sometimes pain persists even after the initial injury is healed.
That's a great question. And it turns out that chronic pain conditions often involve dysregulation of TRP channel activity,
Meaning?
meaning the channels can become either overactive or they can malfunction, and this leads to persistent pain signals.
So even when there's no longer any actual danger, these TRP channels keep firing, sending those pain signals to the brain.
Exactly. And that's one of the big challenges in treating chronic pain, figuring out how to reset these TRP channels so they're not constantly sending those pain signals.
That makes sense. So I'm guessing researchers are trying to figure out ways to target these channels for pain relief.
Got it. By developing drugs that can block or modulate the activity of specific TRP channels, we could potentially create more effective and targeted pain therapies.
That's amazing. So instead of just masking the pain, we could actually address the underlying cause.
Precisely. It's a really exciting area of research.
Wow, we've covered a lot of ground already. We've talked about how these TRP channels are triggered by all sorts of different stimuli,
Yeah.
the many different types that exist,
No.
how their sensitivity varies,
Right.
and their crucial role in both pain perception and protecting us from harm.
It's a lot to take in, for sure.
But it's fascinating stuff. I'm starting to see why you're so excited about these TRP channels.
Oh, there's so much more to talk about. For example, we haven't even touched on how these TRP channels are regulated and fine-tuned by the body.
Hold on. If these channels are so sensitive and react to so many things,
Yeah.
how do they not just fire off constantly? Our senses would be totally overloaded.
That's a great question. And it gets to the heart of how our bodies maintain balance. There are actually several intricate mechanisms.
Oh, this sounds complicated.
It is, but it's really cool. There are these different molecules and processes that interact with TRP channels to either enhance or inhibit their activity.
So it's like a delicate balancing act.
Exactly. It's how our bodies make sure that these TRP channels are responding appropriately to stimuli without going into overdrive.
This is blowing my mind.
And we'll dive into all those fascinating mechanisms right after the break.
Okay, so you were saying there are all these mechanisms that keep our PRP channels from, going haywire.
Right. One of the key mechanisms is called phosphorylation.
Phosphorylation. Okay. Remind me what that is again?
Sure. It's basically a process where a phosphate group, it gets attached to a protein.
Attached, like it's a sticky note or something.
Yeah, kind of. And this sticky note, it changes how that protein behaves.
So in the case of TRP channels.
Yeah, with TRP channels, phosphorylation can either make them more sensitive to stimuli,
Okay.
or it can make them less sensitive. It's like a dimmer switch for our senses.
That's pretty amazing. So it's like our bodies have this built-in system for fine-tuning our sensory experiences.
Exactly. And phosphorylation is just one of the mechanisms.
There are more.
Oh, yeah, there's a whole bunch of them. It's like a whole orchestra of regulatory mechanisms, all working together to keep our senses in balance.
An orchestra. So what are some of the other players in this TRP channel orchestra?
Well, one really interesting example is a group of proteins called agouti-related proteins, or AGRPs.
Agouti-related, like those little rodents with the cool coat patterns.
That's right. The name comes from the agouti gene, which is what gives them those distinctive color patterns.
So what do these agouti proteins have to do with TRP channels?
Well, it turns out they play a role in regulating the activity of a specific TRP channel.
Which one?
The MC1 receptor.
The MC1 receptor. Hold on, that sounds familiar. Isn't that involved in pigmentation, like skin color and stuff?
You got it. The MC1 receptor is a key player in the pathway that produces melanin.
Melanin, right? That's the pigment that gives our skin its color.
Exactly. So how do these agouti proteins fit in? Well, they actually act as an antagonist to the MC1 receptor.
Antagonist.
Yeah, it basically means they block the receptor from doing its job.
So they prevent the MC1 receptor from producing melanin.
Not exactly. They block the MC1 receptor from being activated by other molecules.
Other molecules, like what?
Like a hormone called MSH.
MSH.
Yeah, it stands for melanocyte-stimulating hormone.
Okay, so MSH stimulates melanin production.
Right. And when MSH binds to the MC1 receptor, it triggers a whole cascade of events that ultimately lead to the production of melanin.
But the agouti protein gets in the way.
Exactly. The agouti protein binds to the MC1 receptor and prevents MSH from binding.
So it's like a competition between MSH and the agouti protein.
Yeah, you could think of it that way. And this back and forth, this competition between MSH and agouti, is actually what determines those cool coat color patterns in agouti rodents.
Wow, that's amazing. So we've got these agouti proteins, which are involved in coat color.
Right.
And they're also regulating the activity of a TRP channel. which is involved in all sorts of sensory experiences.
Exactly. It's a really cool example of how seemingly unrelated processes can be linked at the cellular level.
It really highlights how much we still don't know about the complexity of these systems.
Absolutely. And it makes you wonder what other connections we might find as we continue to explore this world.
It's pretty mind-blowing stuff. So we've got phosphorylation, agouti proteins, MSH, all these intricate interactions happen.
Yeah.
It's like a whole hidden world operating beneath the surface of our everyday experiences.
And understanding this hidden world is incredibly important because it could lead to some amazing therapeutic applications.
How so?
Well, if we can figure out how to precisely modulate these TRP channels and all the regulatory mechanisms,
Right.
we could potentially develop targeted treatments for a wide range of conditions, not just pain, but potentially all sorts of things.
That's really exciting. It's like opening up a whole new toolbox for medicine.
Exactly. And speaking of discoveries, did you know that TRP channels were actually first discovered in a pretty unexpected place?
Really? Where?
It all started with a frog.
A frog. Really? What kind of frog are we talking about here?
It's a type of African clawed frog called Xenopus laevis.
Xenopus laevis. Why does that name sound familiar?
I think they use them a lot in research.
Oh, right. They're like a model organism or something.
Exactly. They're really useful for studying all sorts of biological processes.
Including TRP channels.
Yeah. Well, it all started back in the 1980s. These researchers were studying the visual system of Xenopus laevis.
So they were trying to understand how frogs see.
Exactly. And they were specifically looking at how light triggers electrical signals in the frog's retina.
Okay.
And they noticed that when they shone a bright light on the frog's eye, there was this specific type of cell in the retina that produced a unique electrical response.
Wow.
Yeah, it was really interesting. And so they wanted to figure out what was causing this light-triggered signal.
So what did they do?
Well, they started by looking at the cell's membrane. They thought there must be some kind of protein there that was responsible for this light sensitivity.
A protein. Makes sense.
Yeah, proteins are basically the workhorses of the cell, right? But they carry out all sorts of important functions.
Right.
And in this case, the researchers figured there must be a protein that was acting like a gateway, allowing ions to flow into the cell when it was stimulated by light.
I see. So the flow of ions creates the electrical signal.
Exactly. So they spent years, literally years, studying this. And finally,
They found it.
they found it. They identified a brand new protein that they named TRP1.
TRP1, the first TRP channel.
Yep, that's where it all began. In the eye of a frog. Pretty amazing, huh?
It is. So this was a huge discovery, right?
Well, yeah, it totally changed the game.
Yeah.
Because scientists realized that TRP1 wasn't just some weird quirk of frog vision.
It was something bigger.
Way bigger. They figured it probably represented a whole family of proteins, all involved in sensory perception.
Across different species.
Exactly. And they were right.
So this accidental discovery in a frog's eye ended up unlocking this whole new world of sensory biology.
It's pretty incredible, right?
It is. So we've gone from the burn of a chili pepper
Yeah.
to the cool sensation of mint,
Uh-huh.
from the intricate dance of phosphorylation and agouti proteins,
Right.
all the way back to the groundbreaking discovery of TRP1 in a frog's eye.
Yeah.
It's been quite a journey.
It has. And we've only just scratched the surface. There's still so much we don't know about these TRP channels. But one thing's for sure, they're absolutely essential for how we experience the world.
Well, I have to say, I've learned a ton today. I never realized how much was going on beneath the surface of our everyday senses.
It's a whole hidden world down there. And who knows what we'll discover next. Maybe we'll find new ways to treat pain.
Or enhance our senses.
Or even create entirely new sensory experiences.
Now, that's a thought-provoking idea. Thanks for taking us on this deep dive into the world of TRP channels. I think I'm going to be looking at chili peppers and mint in a whole new way from now on.
My pleasure. Always happy to share the wonders of cellular biology.